Welcome! In this video, we're going to provide an overview of the logics behind two of the most widely used univariate methods to analyze the EEG signal, event-related potentials and time-frequency analysis. Having these notions will be important to later on understand the contributions that multivariate analysis have brought to the field. We will begin by talking about the need to reduce the dimensionality of the EEG signal and how univariate analysis have approached this. We'll next briefly outline the logic behind ERPs and frequency analysis, some of the underlying assumptions and classic inferences made from these data. The brain is a complex system, and when we measure the electrical activity that it generates by placing electrodes on the scalp, we obtain a complex signal as a result. At any given time, there are multiple neural assemblies generating equivalent dipoles that summate and reach the scalp through volume conduction. All of this is registered as rapid changes in voltage, taking place over many electrodes. The strategies used to link such complex set of data with specific cognitive processes are not straightforward, and it took researchers several decades to come up with feasible solutions. First, we need well-designed experimental paradigms that isolate and manipulate the cognitive processes of interest, a tradition that started in cognitive experimental psychology with Donders. But also, we need to reduce the dimensionality of the problem posed by the complexity of the EEG data. That is, we need to find summary dimensions that extract the important information and disregards other sources of variability. To do so, univariate analysis focuses on a single dependent variable at a time. In EEG, classic analysis on this line focuses on the electrical activity related to task events in an average of electrodes. These can be analyzed in the time domain with event-related potentials or in the frequency or time-frequency domain. For event-related potential analysis, the stimuli of interest are presented a large number of times to obtain enough observations to be able to extract their evoked activity. The segments containing these stimuli are later epoch and afterwards averaged, resulting in a series of positive and negative peaks that have different labels according to convention. Take into account that the resulting ERP results from heavy data averaging, as it is a reflection of a large number of trials put all together. When we average EEG locked to a stimulus, we obtain a sequence of negative peaks. Remember that their polarity depends on the orientation of the dipole generation in relation to the scalp surface, and not to the excitation or inhibition of cognitive processing. These peaks are characterized and labeled according to their polarity, timing, and typical topographical distribution on the scalp. It is common to display ERPs also in topographical maps, as you see in this figure displaying the N2 and P3 potential, as this facilitates to grasp their topographical distribution in specific time points. In contrast to the ERP peaks, which are observed in the waves obtained, ERP components are inferred from parts of the waves or its peaks. Components have been associated by previous research with specific cognitive processes. The task that participants perform modifies brain activity, and this affects the amplitude and timing of their scalp projected waves and peaks. But these observed waves and peaks reflect the combination of multiple underlying brain sources. For example, in this image you have 3 neural generators or dipoles represented in red color, and 3 recording scalp electrodes in yellow. You can see how each of these sources generates positive and negative voltages that change over time. However, this is not what we see with the surface electrodes. Each of the 3 signals we obtain in our EEG reflects a summation of the polarities of these 3 different sources with a weight that depends on various factors, including the relative position and distance from each other and from the recording electrodes. Take a look at how the source activity differs from the signal that we are able to see in each sensor of the scalp. So, taking into account the complexity of linking inferred components with the peaks and waves we observe in our ERPs, how do we manage to use these markers to study human cognitive functions? For over decades, research has accumulated an extensive literature linking components with specific cognitive processes. Usually, researchers focus on specific peaks because they are interested in the cognitive functions associated with each of them. They perform their experiments and investigate how their manipulations affect the amplitude and peak timing. They may look at the P1 and N1 if they are interested in perceptual processing, at the N170 if they are using phases, at the N2 if they want to study cognitive control, or at the N400 for semantic processing. However, there are some aspects of this process worth highlighting, especially because multivariate analysis sidesteps these problems. First, how should we isolate or select the specific peaks or waves that reflect our component of interest? That is, how we do decide which specific electrodes and time windows to use. And second, how much can we trust the reverse inferences that we extract when we observe differences in the peaks and waves that we have analyzed? So, how to isolate components of interest? Imagine that you run your experiment with a 64-channel EEG set and you obtain something like this for your ERPs. If you wanted to study, let's say, the N1 and the P1, which electrodes would you pick? All occipital and parietal one? What about the temporal electrodes? Would you include them, or only a subset of these? Also, which specific windows of analysis will you use for each of them? 100 to 120 milliseconds of the P1, for example? Ideally, all of these details should be clearly specified in a pre-registration of the study before the researcher looks at the data, as this looking in advance is a potential source for experimental bias. However, a specific pre-registration of these aspects of an ERP experiment may prove difficult in many cases. This is because particular task manipulations in each experiment may change the specific topography and timing of the peaks and waves in the ERPs, and it is very hard to predict these slight changes in advance. Because of this, ERP analysis usually begin with the researcher defining the topography of the peaks of interest, that is, the electrodes and temporal windows to enter in the subsequent statistical analysis. And to do so, the researcher is guided by previous literature and many times by visual inspection of the data to be analyzed. Hopefully, you can see how this brings a certain degree of ambiguity in the process that may potentiate the a priori biases that the researcher may have. And also, very importantly, if the data to be analyzed is also used to select a subset of the same data, the study incurs in a circular analysis, which inflates the rate of false positives. This is because the assumptions allocate part of the error variability present in the data to the experimental manipulation, and the results observed reflects a mixture of these two factors. This is also known as double dipping, and every researcher should be very careful to avoid this circularity, as it has a strong impact on the reliability of the conclusions extracted from the experiment. Another issue is the reliability of the inferences that we make from ERP components. There is extensive literature linking components with specific cognitive operations, such as the N2 with response inhibition in the context of cognitive control. However, taking these peaks as index of these associated processes is a slippery road. This is because the inferences made in this direction are only as solid as the empirical evidence that has linked the component with the cognitive operation and the specificity of such link. This issue has been extensively debated in the field of fMRI research, where the reverse inference fallacy is a rather common phenomenon in the literature inferring cognitive processes from brain activation patterns. To provide an example, think about the following reasoning. If I am Granada, then I am in Europe. As it happens, I am in Europe, and therefore I conclude that I am in Granada. It is obvious to all of us that the conclusion, therefore I am Granada, is not valid, as there are many other cities that also fulfill the condition of being in Europe that are not Granada. Now, let's have another reasoning using an ERP component instead. As it happens, previous literature has repeatedly shown that if participants inhibit responses, then the amplitude of the N2 component is heightened. I can see that my experimental manipulation has increased the amplitude of the N2 component in a significant manner. Therefore, I conclude that inhibition processes must be involved in my experimental manipulation. Again, the structure of this reasoning is not logically valid, as there may be many other cognitive processes that may also affect the N2 amplitude apart from response inhibition. So, when making inferences from the effects observed in ERPs, we have to keep in mind that we can trust reverse inference as much as there is solid evidence linking the components with the cognitive operations with specificity, that is, without alternative potential explanations for the observed effects. Okay, now that we have talked a bit about ERPs and its associated analysis, let's very briefly overview the analysis on the frequency domain. The EEG signal contains activity that is systematically time-blocked to stimuli and that appears with a fixed latency. EEG is averaged into ERP to extract this evoked activity, which relates to the components. However, the EEG also reflects loss of brain activity that is task-relevant, but it is not precisely time-locked to the stimuli. Because of this, the so-called induced activity is lost during the averaging processes, and thus we cannot study it with ERPs. However, induced activity can be studied performing frequency analysis. In frequency analysis, the EEG signal is transformed from the time domain to the frequency domain. Using the Fourier transform, the complex signal within a time window is decomposed into different frequencies and the power of each of them, as you have displayed in the graph. This analysis, however, lacks temporal resolution and thus ignores one of the assets of the EEG. To recover the temporal information and be able to study how frequencies change in time, wavelet convolution is frequently used. This way, time-frequency analysis allows studying the oscillatory activity evoked by and induced during information processing in the brain, with high temporal resolution. Analysis of time frequency have been very useful to advance our understanding on human brain function. Among other things, this approach increases significantly the types of analysis that can be applied to EEG data, and extend the scope of the theoretical questions that can be explored. However, the risks, as we discussed in previous ERP research, are also present here. Researchers should be careful to avoid circularity in their data analysis practices. Reverse inferences are also dangerous in this field. For example, the fact that alpha activity has been linked with inhibition of information processing does not necessarily mean that every alpha that we observe in the scalp indicates the presence of such inhibition. To finalize, a few take-home messages to close the video. First, univariate analysis proposed various approaches to reduce the dimensionality of the complex data we measure with EEG so it can be associated with specific cognitive processes. The field has significantly advanced our understanding of how the brain works by using ERP and time frequency analysis. Importantly, researchers have to pay close attention to avoid circularity in data analysis and also to the reverse inferences made with the data obtained. Luckily, the use of multivariate analysis that we will see in the next video may be handy with this. For additional resources, here you have links to 2 different online courses on time frequency and ERP analysis, and also to review papers and a short book that extend the contents we just covered.